Welcome to this presentation on the Ziehl-Neelsen staining technique. In this session, we will go through the step-by-step procedure and its important applications in medical laboratory practice. Ziehl-Neelsen staining is a special staining technique used to identify acid-fast organisms, particularly mycobacterium species such as Mycobacterium tuberculosis. It is widely used in the diagnosis of tuberculosis and other chronic infections. The principle of Ziehl-Neelsen staining is based on the presence of mycolic acid in the cell walls of certain bacteria. These waxy lipids make the cells resistant to decolorization by acid alcohol. As a result, acid-fast bacteria retain the primary stain and appear red, while non-acid-fast organisms lose the stain and take up the counter stain. The materials required include a clean glass slide, an inoculating loop, a Bunsen burner, absolute alcohol, carbylfuchsin as the primary stain, acid alcohol as the decolorizer, methylene blue or malachite green as the counter stain, and a microscope with oil immersion. Place a small amount of specimen onto a clean glass slide using a sterile loop. Spread evenly to form a thin smear and allow it to air dry completely. A properly prepared smear ensures accurate staining and observation. Pass the air-dried smear through a flame 2 to 3 times or cover with absolute alcohol for at least 2 minutes or until completely evaporated. This kills the organisms and fixes them onto the slide, preventing them from being washed away during staining. Flood the slide with carbylfuchsin and gently heat until steam appears. Allow the stain to act for 3 to 5 minutes, ensuring the smear does not dry out. This step allows the stain to penetrate the waxy cell wall of acid-fast bacteria. Gently rinse the slide with clean water to remove excess stain while preserving the stain smear. Apply 3% acid alcohol to the slide until no more red color runs off. This step removes the stain from non-acid-fast bacteria, while acid-fast organisms retain the red color. Proper timing is essential for accurate differentiation. Rinse the slide again with water to stop the decolorization process. Apply methylene blue or malachite green for about 1 minute, then rinse and allow the slide to air dry. This stains non-acid-fast bacteria blue, providing contrast for easy identification. Examine the slide under a microscope using the oil immersion objective. Carefully observe the stained cells for their color, shape, and arrangement. Acid-fast bacteria appear as bright red or pink rods against a blue or green background. Non-acid-fast organisms appear blue or green. This clear contrast allows easy identification of acid-fast organisms. Ziehl-Neelsen staining is primarily used in the diagnosis of tuberculosis. It is also used to detect other mycobacterial infections and plays an important role in research and public health screening programs. Common errors include overheating the slide, which can distort cells, and under-decolorization, which may lead to false results. Other issues include thick smears and insufficient staining time, all of which can affect accuracy. In conclusion, Ziehl-Neelsen staining is an essential technique for detecting acid-fast organisms. It plays a critical role in the diagnosis of tuberculosis and other infections. Mastery of this method is vital for laboratory professionals. Drop your questions in the comments section below. Remember to like the video and subscribe to the channel to enjoy more exciting videos.